Good morning, Professor. Myself, Mehak Vora, and my partner, Yashvi Patel, we are here today to discuss about the experiment that we have performed in Micro Project 2. Our experiment is all about detecting the antimicrobial activities of two fruits, which are pomegranate and orange, against three organisms. One is gram negative, one is gram positive, and one is fungi. The organisms that we are using are S. aureus, P. aeruginosa, and Candida albicans. We are detecting the antimicrobial activities not of the fruit but the fruit peels in this experiment. All the experiment in this scientific analysis has its own importance. Our experiment is also very important because nowadays human health is the most important concern, is the most important topic to talk about. Our human health is mostly affected by the food that we are consuming. This food nowadays are consist of some artificial chemicals which are used to preserve the food. This chemical includes the use of benzoates, sulfates, nitrates, which are very harmful to humans if they are used in the long terms. Nowadays, these foodborne illnesses are caused by some pathogenic bacteria which include Escherichia coli, P. aeruginosa, C. albicans, S. aureus, etc. These are the common bacteria that are found in the nature. To eradicate the use of the chemical and artificial preservatives which are harmful, we are here with a new term of natural preservatives. So we are using natural peels of pomegranate and orange as preservatives for our food, and we are detecting the antimicrobial activities of these fruit peels to eradicate these organisms. In these slides, we have mentioned some important materials that we are going to use in our experiment. The three purity plates are used, which are already inoculated with the particular bacteria that we needed. Oven is used for drying of the peels. Shaker is obviously used for shaking the solvent along with the peels to make the extract. Extraction funnel and Whatman filter will be used in the extraction process. Ethanol is used as the main solvent for our experiment. We haven't used methanol because it is toxic. Amateur plates are the plates that we will be used during well and disk diffusion. 10% DMSO will be used as control. Micropipettes, we are using micropipettes for suspending the liquids in the wells during well diffusion. Sterile swabs are used to make the lawn of the bacteria, and pasture pipettes, the second hole of the pasture pipettes is used to make wells in the disk. Our experiment is divided into three parts. First is the plant material extraction and suspension. Second is tricking for the purity plates and microbial suspensions. Third includes antimicrobial studies using disk and well diffusion technique. Now I am going to explain the whole method that we have used to prepare the extract. Firstly, we have purchased fresh pomegranate and orange from the market. We have washed it thoroughly and sterilized it by washing it with distilled water. Then the peels were removed from the fruits, and for the preparation of the powder, we dried the peels at 40 degrees Celsius for 24 hours in oven. Then they were grounded using mortar and pestle. The peels powder was stored at 4 degrees Celsius. After storing the peel powder for 2 days, we kept them for shaking with ethanol solvent for 3 days, and the measurement that was used was 25 gram of powdered peel with 225 ml of ethanol. The peel then were filtered and extracted by using Whatman filter and extraction funnel. Then they were put in the fume hood for evaporation. This evaporated and extracted material will be used for our further process.
Hello everyone, my name is Yashvi. I am going to discuss the next steps in the experiment. So the next important step in the experiment is media preparation. For this experiment, we need both well and disk diffusion. The best suited agar for it is MHA. The media was prepared with the instructions provided on the media bottle. The next step was streaking for purity plates. We streaked our three organisms on TSA. The organisms were S. aureus, P. aeruginosa, and Candida albicans. The plates were inoculated for 2 days and at 35 degrees Celsius. Moving on to our final test. Firstly, for the disc diffusion technique, we have used our blank disc and our antibiotic disc, specific for both gram-positive, negative, and fungi. Firstly, the plates were swabbed with the bacterial lawn. Each one was kept with a blank disc having orange and pomegranate extract peel and one antibiotic disc, which was our positive control. The antibiotic discs which were used are tetracycline and chloramphenicol. We used tetracycline for both our gram-positive and negative bacteria, while chloramphenicol was used against fungi. For the well diffusion, we have firstly swabbed the whole plate with our bacteria, and then using a cork borer or a pasture pipette, we can put holes inside the plates in which we are going to put our extract and also our negative controls. For the extract, we have put both our pomegranate and orange peels. We have put negative controls, which was 10% DMSO and ethanol. The plates for well diffusion and disc diffusion were incubated at 35 degrees Celsius for 24 hours. This slide showcases the results for our well diffusion, and clearly we can see that the pomegranate showed a highest and clear zone of inhibition around itself. For pomegranate peel extract, the highest zone of inhibition is seen at 4.6 cm for Pseudomonas, and the next was seen for Candida albicans at 4.5 cm. Pseudomonas aeruginosa showed greatest zone of inhibition than S. aureus and Candida albicans. Both are positive controls showed growth, as ethanol and 10% DMSO cannot inhibit the growth of our bacteria. The next slide shows the results for the disc diffusion. It also supports the results for our well diffusion, as we can clearly see that pomegranate peels shows the highest zone of inhibition. It is most effective on P. aeruginosa. Here, the antibiotic discs were negative controls, as the antibiotic discs which were used were tetracycline and chloramphenicol, which always shows no growth around themselves. P. aeruginosa showed 5 cm of zone of inhibition for pomegranate peels, while for the antibiotic disc tetracycline it only showed 4.5 cm. Thus, it confirms that pomegranate and orange peels have antimicrobial activities and effects that can be used instead of artificial preservatives. The next step in the experiment was performing MIC, minimum inhibitory concentration. It was performed where we used 1 drop of our organism P. aeruginosa inoculated in a TSB broth and our diluted pomegranate peel extract. The growth was seen after 1/1024 dilution at the last two dilutions, so 1/1024 becomes our MIC for the growth of P. aeruginosa against pomegranate peel extracts. The hypothesis of our experiment is hence proved. We can conclude that both pomegranate and orange peel extracts have antimicrobial properties and are effective on our organisms, while pomegranate is more effective than orange peels on our organisms. Thank you.